Thank you very much, Bob, for that introduction and the opportunity for introducing Alcyonics assays and how we have repurposed them for COVID-19 research. Oops. First of all, I'll introduce you to Alcyonics and the Skimmune technology platform. Alcyonics is a spin-out company of the University of Newcastle, and it's based in the northeast of England. It's a small biotech, has been trading since 2009, and it's a service provider providing unique tests for preclinical testing, product safety testing, using human in vitro skin tests. We have over 20 years experience in skin explant models and in vitro models of disease. They're non-animal alternatives, and we support the National Centre for Replacement, Refinement and Reduction of Animals in Research. We can identify adverse immune reactions to drugs before you take your product into the clinic. Using a Skimmune skin explant human model is highly predictive of adverse responses in the clinic, and it can be applicable to all types of drug types: small molecules, biologics, nanomedicines, oligonucleotides, and cellular therapies. The Skimmune assays and platforms uses human blood samples and a small skin biopsy. The biopsy size is about 4 millimeter, and it's the same size of biopsy that you would take if you had a skin disease and you were at the dermatologist. It's a very small biopsy. We've used our assays for pharmaceuticals and small molecule drugs, and we've worked with Pfizer recently and published in 2019 with them for the assessment of small molecule drugs. This is the type of assay that can be repurposed for COVID-19. So first of all, we take a blood sample from a healthy volunteer. We isolate the CD14 positive cells and we generate dendritic cells. Those dendritic cells are activated by the compound of interest, the small molecule drug. We then add back autologous T cells and activate those T cells with the combination co-culture. Development of, we then look at the proliferation of the T cells and cytokine and chemokine release, and then those autologous sensitized cells are added back to the small piece of skin. And the skin then forms damage within that, in the skin due to the compound immune reaction, and this is shown in the next slide. So the results give rise to a grading due to the test therapeutic from a grade 1, which is negative, where the dermis and the epidermis is normal. It's a negative response. It's the type of response which occurs with media alone or autologous cells alone within the assay. Then grade 2 to grade 4 are positive reactivity. In grade 2, you have vacuolization within the epidermis. It's a little bit like an in vitro rash. Grade 3 is where you get subepidermal cleft formation, where you get separation of the dermis and the epidermis, forming in vitro blisters in the skin. And grade 4 is when you get complete separation of the dermis and the epidermis and the skin begins to fall away. All of these reactivities are clinical grades and are used as a clinical grading system within the clinic. They're also predictive of systemic disease, not just skin reactions. The grading process is performed by 2 independent professionals trained in this grading process. Apologies, that's been turned off. The screening assays are T cell proliferation and interferon gamma assays. They are where we test the T cells after interaction with the compound and isolation of interferon gamma. The assays were validated up using sensitizers and non-sensitizers. On the left-hand side of the graphs, you can see T cell proliferation of positive sensitizers, and right-hand side, the negative non-sensitizers. And you can see the variation that you obtain with this type of assay. Within Skimmune, the skin damage actually is more sensitive, and you don't need so many donors to show that you've got a positive or negative response with the compound. In response to the global effort towards COVID-19, Alcyonics have repurposed their human-based assays to aid efforts and study the immune response to the virus, identify new therapeutics towards SARS-CoV-2, and expedite the safety and efficacy assessment of new vaccines, drugs, and repurposed drug candidates. First of all, a little bit of the background of the immunopathology of COVID-19. The incubation, as you know now, probably is of COVID-19 SARS-CoV-2 vaccine is between 3 and 7 days. 80% of infections are mild or asymptomatic, 15% are severe, requiring oxygen, and 5% are critical, requiring ventilation. It uses the ACE2 angiotensin converting enzyme 2 receptor as a host receptor to infect human airway epithelia, but this is also present on cells of the heart, arteries, kidneys, intestines, giving rise to the type of disease parameters which are being seen in the patients. Spike and nucleocapsid protein are the most immunogenic expressed proteins during infection, and specific T-cell responses against SARS-CoV-2 is important for recognition and killing of infected cells, particularly in the lungs. The cytokine storm produced by SARS-CoV-2 in susceptible individuals is a severe systemic inflammatory response, releasing pro-inflammatory cytokines such as the interferons and interleukins and chemokines. Differences in levels of the chemokines and the cytokines when comparing severe versus mild infected patients may allow indications of susceptibility. So I'll now describe some of the COVID-19 assays that we have repurposed. Some of the questions you might want to ask is, do you need assays to test immune responses to vaccines or novel therapeutics? And are you looking for new drug targets? Do you require safety and efficacy testing for COVID-19 vaccines under development? Do you require testing of your immunomodulatory inhibitors for SARS-CoV-2 immune responses? Do you want to test new or repositioned drugs to see their effect on SARS-CoV-2 immune responses? The first aim, which I'm going to describe, is evaluating immune responses to the SARS-CoV-2 peptides, vaccines, and novel therapeutics critical for the management of patients in clinical trials. So in order to repurpose our assays, we of course take this blood sample from the healthy volunteer, and we can test the isolated dendritic cells with the vaccines or the peptides, activate them with the DCs and the T cells, and generate cytokine and chemokine profiles by isolating the supernatant and looking at proliferation assays. We then get an idea of the Th2, Th1 type profiles of the vaccine and the peptides, and give you an indication of the immune pathways involved. We can also look at mRNA expression analysis, which I'll show in a little bit more detail on the next slide, in the way of looking for new drug targets. So if we want to test COVID-19 peptides identified for spike and nucleocapsid immunogenic proteins, which has been associated with class 1 and class 2 epitopes to discover novel drug targets. We can assess the immune response to the peptides via the assay I've just described and use HLA-typed human PBMCs. We can assess the cytokine profiles and chemokine profiles and look at the DC and TC interactions by RNA-seq, RT-PCR, ingenuity pathways, and bring together epidemiological factors, HLA genotypes, ABO phenotypes, and develop disease structures, disease signatures for drug discovery and high and low responder profiles. This will then inform the cytokine storm response, pathways of immune activation, differences in susceptibility to COVID-19, and validation of studies are on the way in COVID-19 patient samples. This gives valuable information for drug discovery efforts to design therapeutic strategies to understand the immune response and aid in vaccine novel therapy development. What about safety and efficacy testing for COVID-19 antiviral T-cell therapy under development? We have used this technology to look at cytomegalovirus-specific T-cells isolated by interferon gamma secretion assays, and this work was published in 2016. In these assays, we look at the antiviral T-cells which are being developed by use of the peptides, development of cloning of T-cell for T-cell therapy, and we assess whether those T-cells are specific or not by use of allogeneic assays and T-cell specificity assays with readouts of antiviral specific T-cells, such as ELISA spot or secretion assays for interferon gamma, and SCIMMUNE assays, which give histopathology readouts. These assays indicate whether how specific the antiviral T-cell is for a clinical trial, and also how efficacious they are in development of the antiviral T-cell therapy. What about testing of your immunomodulatory inhibitors to SARS-CoV-2? Here we can modify our assays for this within the SCIMMUNE platform and determine the efficacy of the immunomodulatory drug. We can start with a mixed lymphocyte reaction between two non-compatible individuals, and in 9 out of 10 cases we always get a grade 3 reaction in the skin, which is a severe reaction, and this can be downregulated by the presence of antibody or inhibitor to the immune response to the SARS-CoV-2 virus, and it can also be a dose response. So a grade 3 reaction can be downregulated to a grade 2 and a grade 1 reaction in a dose response manner, and you can look at the efficacy of your immunomodulatory drug in a very sensitive manner for efficacy of your inhibitor. So in summary, working with Alcyonics, do you need assays to test immune responses to vaccines or novel therapeutics? Our standard patented SCIMMUNE platform is suitable for the testing of immune responses to those vaccines and novel therapeutics. Are you looking for new drug targets? This is an exciting development within Alcyonics in that we can induce immune responses, investigate the pathways and networks involved to identify drug targets by RNA-seq and mRNA expression and transcriptomics, multiplex cytokine chemokine profiles, and investigate the mode of action and take into consideration HLA, ABO phenotypes, as well as epidemiological factors which are important in assessing the immune response to COVID. Do you require safety and efficacy testing for COVID-19 antiviral T-cell therapies under development? Using T-cell specificity assays in our SCIMMUNE platform, we can demonstrate safety and efficacy of the antiviral vaccines. Do you require testing of your immunomodulatory inhibitors for SARS-CoV-2, or do you want to test new or repositioned drugs to see their effect on the immune responses? To assess the efficacy of your drugs to suppress the cytokine storm, Alcyonics can use the immunomodulatory immune inhibition SCIMMUNE model, as well as inhibiting T-cell responses in the assessment of drugs. Thank you very much for listening. I hope you have enjoyed an overview of Alcyonics. If there's any further questions, please get in touch with me via email, and please type out.
Today, I'd like to talk to you about some of the services and resources that we've been working with for COVID research at Bepracell. First, a little bit of background about our company. We were founded in 1989 as BioServe Biotechnologies Limited. And we were started by 3 of us who are all still here 30 years later. We all worked at NIH together, and in 2007, we acquired a large biorepository, and at that point we realized that that was a wonderful resource, but it didn't have everything that everybody needed. So we decided to begin prospective collection studies, and we started working with the IRB to get our proper paperwork and all the protocols and the informed consents. And we've been doing prospective collection since 2007. In 2014, Reprocel Inc. of Japan acquired BioServe, and we merged with StemGen to add some stem cell services and products. And we were rebranded as Reprocel USA. And in 2019, just last year, we've gained ISO 9001 certification. So just a little bit about our IRB approved prospective collections. Currently, we have 6 collection sites that we work with, including our own facility. We have partnership with various cancer centers and hospitals using their IRB approved protocols and consents, as well as partnerships with other biospecimen providers. Our biorepository has approximately 600,000 samples of various disease indications. We have serum, plasma, pathology blocks, frozen tissues, DNA, and RNA. And our custom collections, we also collect the same types of material. We have healthy donors for whom we primarily collect blood products, but also disease donors, particularly oncology, infectious disease, autoimmune patients, metabolic disease patients, and we also do inflammatory disease collections. We continue to provide our molecular biology services and other services, which were initially our core products that we had in our core services that we had, which include genotyping, RNA, DNA extraction. As I mentioned, we are working now with stem cell development developing stem cell lines and testing those lines, as well as the stem cell products. We can perform cytotoxicity testing and drug discovery services as well. So on to the COVID. Right now, as of a couple days ago, there were 10.4 million cases worldwide and over 508,000 deaths. In the United States alone, we have 2.6 million cases and over 127,000 deaths. As we all know, right now, the COVID-19 cases are exploding in the US again. As of a few days ago, we have 700 new confirmed cases per million residents in the United States. And you can see in comparison with Europe, Canada, and Japan, we're very much on an upward trend. So we are definitely living in the COVID era. And we have many samples available that are pre-COVID. Anything collected before December 2019 is considered a pre-COVID sample, in which you don't need to test for positivity and you don't need to test for negativity. Everything is negative. However, anything collected after December 2019 needs to be tested both to prove positivity and to prove negativity. The types of biospecimens that we are working with and have the capability to work with are plasma, serum, the nasal swabs, which are used for the RT-PCR diagnosis of the initial infection, There have also been assays designed for saliva, although those haven't had great success yet. Fecal samples have been tested for COVID, as well as urine samples. And those are samples that we would be able to collect. So if we just look for a minute at what we have in our biorepository, we've been getting quite a number of requests for pre-COVID samples. And we have, as far as healthy controls go, we collected samples from more than 12,000 subjects, all of whom we have DNA from. We have serum from over 7,000, and we have more than plasma from more than 1,000 of the patients. In addition, we have whole blood and some PAX gene, or some PAX tubes from which we can isolate RNA. Now, this is the non-oncology portion of our biorepository, and you can see in bold that I indicated that we have pre-COVID samples, particularly serum samples from asthma, cardiovascular disease, pneumonia, deep vein thrombosis, diabetes, and end-stage renal disease. All of these are diseases in which the people that have these diseases seem to be more susceptible to the devastating effects of COVID-19. So COVID-19 pre-COVID-19 serum is available for many of those diseases. When looking at the COVID positive samples, as Anne mentioned, there are both asymptomatic and most of the patients who develop this are asymptomatic or mildly symptomatic. That accounts for probably 80% of the subjects, and then the severely symptomatic accounts for the other 20% or so. So the asymptomatic and mildly symptomatic samples that we've acquired have been collected in a clinic. They've been prospectively collected using IRB-approved consent forms and IRB-approved protocols. We have testing data available on those samples, and we're able to perform testing as well. And we've used case report forms to collect information from the patients directly, or the subjects directly. On the other hand, the symptomatic, severely symptomatic or acute patients are hospitalized patients. We do not have direct contact with those patients. So what we're able to obtain are classified as remnant samples. We cannot do a prospective collection and request certain types of samples from those very sick patients, which certainly makes sense. They've signed a hospital consent form, which allows for release of their samples and also provides consent for their medical studies department to provide data through their electronic medical record. And we do have testing data available from the hospital patients as well. So for the asymptomatic and mildly symptomatic, as I mentioned, these are community dwelling folks. They've had an RT-PCR positive test. They have no symptoms or mild symptoms, and they have not had treatment. The type of samples that we have collected have been serum and plasma. And in addition to that, we have done some longitudinal collections using plasmapheresis. And again, if there's a project that somebody needs to have done, this is customizable because we design the case report form ourselves. And so we can ask for certain information directly from the subject. This is an example of one of the longitudinal plasma recovered subjects. This is a 34-year-old woman who in February had a positive RT-PCR test. She came to the clinic in April 14th, and had her first IgG positive test there. Since that time, she's donated 7 plasma 7 times, and she's been tested for IgG positive and has been IgG positive, and it 6 times or 7 times. We have approximately 150 longitudinal plasma donor subjects available at this point. Some people have donated plasma only twice, and we've had subjects who have donated plasma up to 10 times. And each time the positivity is run to check for the IgG positivity frequently. So the information available from the asymptomatic and mildly symptomatic subjects are what we collect on our CRF, primarily the age, gender, ethnicity, medications, comorbidity. We have information about the onset date of their symptoms, if they have symptoms, and we have the original RT-PCR results and data and any additional RT-PCR testing, as well as the IgM and IgG results and any subsequent testing that they've had. And as far as the symptomatic subjects, we have information about when their symptoms resolve. From the COVID positive biospecimens taken from the severely symptomatic or acute patients, we have over 1,000 patients from whom we have SST serum. And we also have about 1,000 patients from whom we have plasma. We have about 50% EDTA plasma, 25% lithium heparin plasma, and 25% sodium heparin plasma. From some of the catheterized patients, we have obtained urine from about 50 of those patients. And in addition, we have around 1,500, nasal swabs that are positive and 4,000 nasal swabs that are negative. So the type of information that we have from the severely symptomatic patients are the age, the gender, and the testing information. And then some of the additional information that we're able to acquire from the EMR is the admission date, the symptom resolution information, if they've been on oxygen or if they've been on a ventilator, their outcome status, if they have recovered and been released to home, if they're in acute care, if they've gone to hospice care, or if they've passed away. And also, we do have access to all the lab testing that they've had, both the chemical testing and the hematological testing that's been performed as standard of care for these particular patients. This is just an example just to show you some of the hospitalized patients that we have. As you can see, most of these patients are definitely on the older end of the spectrum. We have the the status here. They've been admitted to the ICU or the emergency room, if they are on oxygen, if they've gone into hospice or into rehab. And again, the symptoms, comorbidities, and the initial diagnosis is available in addition to all the testing that they've had. So what can you do with these kinds of samples? Well, certainly everyone has their own research projects and would probably have good uses for these patients. Some of the things that we're thinking of doing are looking at the genomic DNA to do whole genome association studies, looking for increased risk or protection for certain ABO blood types, and increased risk or protection for certain HLA types. Also, to perform some microbiome studies, we could use, investigate the oral microbiome in COVID positive patients using saliva, mouthwash, oral rinses. We could look at the gut microbiome studies using fecal samples from COVID patients. We could look at post-COVID morbidity in stem cell studies by generating stem cell lines from COVID positive patients who have recovered, and also material taken from COVID positive patients while they're alive who have subsequently died. Another thing that we would be able to do would be to examine our pre-COVID samples for the various diseases that have been associated with a severe outcome in the COVID, and compare that to the positive, our pre-COVID samples, to compare that to positive patients with the same disease. So we are in the COVID era, and we want to win the era by doing good research and being safe.